Chairman. Thank you for the introduction. Hello, everyone. Good morning. My name is Lóránd Kelemen. I'm going to talk about our microtools, microstructures that we are developing in the BRC, in the Biological Research Center, which we are not curing or trying to find cures for treatments as before, but we are trying to provide microstructures, microtools that make cellular single-cell research maybe easier or more effective than before. So the outline is shortly introduce how we can use the microtools or for single-cell manipulation. How are they made? What are the means by which we can move them around, grab the cells and study the cells, and eventually I show you three examples that we published in the last year. So single cells, studying single cells has always been important in biology and in life sciences, and it very often involves moving cells around, manipulating them in a microscopic environment. Under the microscope, for instance, you want to look at the cells in a more detailed way, try to study cell-cell interaction, for instance, but some way you have to physically move the cell around or position precisely to observe. And one way of doing that, these microscopic microtools that we have been developing, can be a possible and promising way. Of course, you cannot touch it with your hands because they're small, but these microtools can be and are useful, as we proved, in manipulating and studying cells. Okay, how are these microtools made? So if you want to be really effective in making them, you have to find a microscopic fabrication way. And what we do in our lab is that we are using photopolymers, light-sensitive material, in which we focus a special laser beam, and in the focus spot where we illuminate this photopolymer, the polymer hardens and becomes and it brings about the microstructure. It's very similar to when you go to a dentist and you have a dental filling and the plastic filling where the dentist illuminates with the UV light. It's pretty much the same chemistry behind, so, but we are doing it in a very, very small scale. It's pretty much a 3D printing in a micro world. And what comes out in the end is a structure like this, where you can see the scale bar is 10 micrometer. If you want to compare it with a hair, for instance, it's about 100 micrometer thick. And this, as you can see, the features of this microtool are much smaller than that. So in the 1 micrometer range or even smaller. And this is pretty much the size of many single cells that we can observe. So we size match these structures to the cells. Okay, how can we move these microstructures around, and how can we use them? How can we grab them? Of course, you cannot use your hand again. What we use is called optical tweezers, or laser tweezers, maybe another word. And it works the following way: you have a laser beam. Again, you focus down to a small spot, and microscopic objects, which the refractive index of which is larger than the surrounding, which is usually water, can be trapped or are trapped in this focal spot. These can be single spheres to begin with, or more complex objects, or even cells can be trapped with a laser tweezer. And the force that the laser tweezer can apply is this small, so 0.1 to 100 piconewtons, 10 to the minus 12 newtons. And in comparison, again, this is this many times. Look at the zeros. How much is small? How many times it's smaller than the weight of a person? But this force range is actually relevant in the dimension of cells or molecules. So that's why the laser tweezer, the optical tweezer, was really powerful and efficient tool to study molecular interactions and cellular interactions in the past. And here's an example of how our micro tools can be moved with the laser tweezer. So imagine that the 4 spherical parts of this micro tool is trapped with 4 individual focal spots, and when we move the focal spot around, of course the small structure will follow its movement. Now it's just a simple rotation back and forth of the microstructure, but we can do more complex motions than this. You can of course use a laser tweezer, as I mentioned, to trap a cell directly, but it has disadvantages. For instance, the cell can be heated, which of course causes damage. The trapping position is quite uncertain because the cell is much bigger most of the time than the laser focus. So you're not certain which part, which cell organelle you are trapping in the cell. And the trap is usually weak, so you can act a much smaller force than what you would do with the microstructures. Therefore, if you have a micro tool like this, and you trap it with the laser tweezer, which has at this point, at this case, 3 focal spots. You focus on these spheres, and you start moving around these 3 focal spots. The microstructure can move around, and if you attach a cell to this microstructure, then of course the cell can move around with the microstructure as well. So you practically add a handle to the cell. And thereby you don't illuminate the cell with the laser. The trapping point is certain because at these spheres are the points of trapping, and because of the higher refractive index difference, it's a much stronger trapping. But still, there are 2 disadvantages. So the cells need to be attached to the microstructure, for which the cell need to be treated somehow, which may not be a good idea sometimes, and the cells cannot be released from the microstructure later on. So that's why we developed a next generation of micro tools, which are not rigid as this one, but flexible. So and why we use that's exactly why we use these deformable micro tools, because you don't need to treat the cell with anything in order to be grabbed by this micro tool, and you can release it after the investigation. So therefore we needed to find a photopolymer which is soft enough and which can be deformed with the small force the optical tweezer can act. And the way it is designed, such a micro tool, it has a rigid part which gives it the structure of it, but the key part is this elastic part which we polymerize in such a way that the actual thickness of this is way below 1 micrometer, is about 0.3, 0.4 micrometers, so 300 to 400 nanometers. And we can tune the softness or the elasticity of this flexible part. So if we make it a little bit thinner, it will be more elastic. If we can make it a little bit thicker, less elastic. So the point is that we can tune the flexibility of these microstructures. And these axes are the points of the optical trap. So that's how we can trap them. Okay. And and in this paper that we published last year, we show 3 examples of the application of these flexible or deformable micro tools. One was simply, let's collect cells. Let's imagine we have a sample space with lots of cells, and you're interested in some of them. You want to collect to a certain space, a certain location, and you don't want or cannot intrude with any other with a tool from outside. So we simply add our micro tools. We grab with the practically invisible hand of the optical tweezer these micro tools and collect the cells. The point is that we want to collect such a way that you don't want to squeeze the cells. So it's a very soft or how to say. Okay, so I can't find a proper word now. So the point is that you don't want to press the cells while we are collecting them. You collect, lift it up, a transport to the location and release them. We prepared even a cage for this. And the microtool looks like this. It has a wireframe structure, which is bigger than the cell itself. And this is wireframe because the viscosity of the liquid is high, of course, and you want to translate it fast. So if it was a solid surface, then the force which acts on it while you are transferring would be high. And this is the collection area where we collected the cells. And this is the process of the cell collection. So first you open up the cell, the cell container with the cell collector with the optical tweezer. You move the cell in, you close it, so the cell is now trapped inside. And as you see, when we carry it, the cell is not squeezed, it is freely moving in. Then we release it at the location in this cage, release, move away, leave the cell, and go back and look that, okay, we dropped one cell to the others. Okay, this was one example. The next example is where we do want to hold the cell firmly for microscopic observation. In this way, with this microtool, we grab a cell such a way that with these 6 small pins, we firmly hold the cell with small fluctuation, and when we hold the structure, the whole cell is not fluctuating more than the resolution of the optical microscope. And the key point here is, if you want to observe the cell from various directions, then you have to be able to rotate the cell around, and that's exactly what happened here at the end of this movie. Okay. Let's go here, somewhere here. If you look at it, we can rotate the cell around to any direction. And on this one, you can see you can have a good view of it from one direction, and then you rotate and hold it like this to another direction, and then you can take 3-dimensional images at any direction you want. And the way we prove that it works actually is that we attached microbeads to the cell and imaged the cell with the microbead attached to it from various directions. And after aligning those directions, those different views, and fusing them, you have a much better view of the cell, because it's an inherent problem of the optical microscope that when you image something, then along the optical axis, the image of any object is elongated. But this elongation can be reused or removed completely from the image. So you have the images of these microspheres, the fluorescent microspheres, are actually sphere-like in the third fused image. So this was possible again by grabbing a cell with this microtool, holding it firmly with very small fluctuation, and imaging from various directions. Now the last example that I want to show you is actually using 2 microstructures, because it deals with the cell-cell interaction in a spatial and temporary, very good, very well-controlled manner. So with one microstructure, you hold the cell, you attach the cell to the substrate, and with the other one, you grab the cell and press it against this cell on the surface. And the position where the location with which you press it against, and the time of the interaction, the onset of the interaction can be controlled very precisely. This is the electron micrograph of one of these microstructures. And this shows you how a cell is grabbed with this microtool. So this is the flexible part of the microstructure. Here you grab at this point with the optical tweezers, you open up, you maneuver the cell in, and when you release, you turn off the optical trap, the elasticity of this structure, of this part, again grabs the cell or blocks the cell keeps the cell inside the structure. Okay, and here's the interaction itself. So once you grabbed one of these cells with the microtool, and the other structure is held with on the surface, you move this structure with the optical tweezer, and the other is moved with the microscope stage. So just one is held with the optical tweezer, this one on the right side, and then you can push them against each other, so you can really make an interaction. And in order to prove that they really interact, in this these cells were previously pushed together, and where you can see that after holding them or keeping them together for a few seconds, actually, it's enough for them, their cell membrane combine a little bit, and a cell tether, a small tube is formed between the two cells once you pull it away from the other cell. And not only you can observe these microtubes, but you can actually measure the force with which they are held together with the optical tweezer. And here's the same thing in fluorescence observation. So this is really a short introduction of these microtools. We have been working with them for 2 decades now, and this is our latest and I think most nicest results. So my take-home message is that doing physics, doing optics and material science combined with biology really can bring about beautiful results, and it's a very fulfilling task to work with these microstructures, and actually a useful thing to study single cells. Thank you very much. Oh, by the way. And
thank you very much. If you have questions to Professor Kelemen, then you can ask in the coffee break, because we are now a little bit behind schedule, but I thank you very much and congratulations.